Awesome. Helen, thank you so much again for joining us today.
So for the party people, my name is Helen, she, her pronouns. My background is in biotech and genetic counseling, but I currently work at Fred Hutch Cancer Center as their scientific content liaison. So that's me. So I don't know if any of the colony out there remember our first Helical Whodunit, but we're back for round 2. So this is a scientific phenomena that we are going to explore and then bring it all back to a little bit of a Fred Hutch research as well. And also, I've only been at the Hutch for about 5 months, and my background is in genetics and in biotech research, but it's not in bone marrow transplantation or immunotherapy or a lot of the things that Fred Hutch specializes in. So we are learning together a lot of times. So jump right in, and it's going to be a blast. The Munchkin.
And the Munchkin would like to learn as well.
Right. Like we are learning more and more that DNA is a lot more complicated than we thought, and that our own DNA is a lot more complicated than we thought, which is why there's this saying in genetic counseling. My mom famously got it wrong, and it was so funny the other day. She was like, "What's that saying you have? Like, slap it out." I was like, "What? What are you talking about? What saying for what?" And she was like, "No, for like genetic testing." And it's when in doubt, punch it out. Which means that if you're not sure if someone's had a bone marrow transplant or like a blood infusion and you want to know what their germline DNA or the DNA they were born with looks like, you should take a skin punch biopsy, which is a sample of their muscle and their skin, because those cells are usually going to be the cells of a person.
But that is dependent on access to the patient, right? So if you're doing something like a crime scene investigation where maybe you don't have that, or if you're tracking down someone, it's a little harder to get that. But it's such a cool thing to have that be like almost a cover up, right? That there's that. What is our medical mystery today?
Yeah, so today, I see we're already in presenter mode. Lovely. If we go to the next slide, we will see that we are on case 002, which is we are who we are, or are we? Or are we? So if we go to the next slide, if this seems familiar to folks, that's because this case is actually taken from real life. And we're just rehashing it together because I think it's really cool for folks who might not have heard of it.
Was this at the Hutch, or was this just at like a general, like popular case?
Yes. So we're going to bring it back to the Hutch at the end with a little bit of fun science. But our first story was not at the Hutch. Our first story is just like a general case thing that happened.
Okay.
But it's just crazy. And it did actually take place in Washington State, which is where Fred Hutch is located, but it wasn't affiliated with Fred Hutch.
Nice. And then Laura was wondering, when people get blood transfusions or donated organs, people get tested and the DNA comes up at the donor and not the host.
Yep. Yeah. So that's the same idea as like a bone marrow transplant. So with blood transfusions, if they've received a blood transfusion, I like how every time we talk about blood transfusions, I'm like, blood, you know. When you receive a blood transfusion, if it's just what they call packed red blood cells or pure red blood cells with no white blood cells or platelets or other cell types, typically, that's considered safe to still have blood-based genetic testing because your red blood cells don't have nuclei, and so they don't have DNA, which is pretty crazy. But if there are white blood cells or any immune cells, then those are going to have DNA from the donor. So yeah, you can definitely still see that. And with donated organs, I don't know much about solid organ transplants, but my guess is that it would probably be similar because that donated organ is going to have DNA from the original owner of that organ. And now it's in your body, so it makes sense to me.
Right, yeah. And so it'd probably be free floating, but maybe there's like a density effect to it too. If it's a single organ, then you're primarily having your own blood cells in there.
And also probably the way we're doing things, right? Like if we're karyotyping, which is like actually looking at chromosomes under a microscope versus doing, as you were talking about, like amplifying DNA from like cell-free fragments. The cell-free DNA might be a mix, but DNA that's actually enclosed in a cell that is, still intact and being sampled, that might be more likely to be the patient's host or the original person's host.
Yeah. Okay. So in our test case, if you're not you, but you're me, who are we?
Yeah. Yes. So going back to our test case, if you're not you and you're not me, who are we? Yes. So 2000s, bring yourself back. Low-rise jeans are in. Everyone's straightening their hair. It's dark days. Early 2000s, Lydia Fairchild has applied for government assistance to help take care of her 2 children. In the state of Washington, where this case takes place and where I currently live, and where Fred Hutch Cancer Center lives, you have to submit a genetic test to confirm biological parentage, DNA parentage, before receiving receiving financial support, at least in the early 2000s. And when they ran the sample, the lab told Lydia that there was no way, based on her DNA, she could be her children's biological parent. So the lab then followed up and they did tests, and the tests confirmed that Lydia's 2 children were siblings to one another, full siblings, and that her partner was their biological father, but that she wasn't related to any of them. If we go to the next slide.
So
yeah,
that would be kind of wild then, right? that already on its own doesn't really make sense.
Yeah.
It's like the joke of like, how does something like that even happen, Munchkin? We're going to figure it out.
Exactly. That's what we're going to explore. So if we go to the next slide, and then, perfect. So we'll pause for a minute. And then I've gone through, I made my best educated guesses for here's what I would think it was. And then sort of made some slides for that. But I'm curious what theories you guys have. One theory, one of the first ones I had was, what are IVF? So maybe Lydia and her partner decided to conceive using IVF or in vitro fertilization. The way that IVF works is you take the sperm, you take the egg, and you combine them in a petri dish in a lab. You get the sperm to fertilize the egg, and you have your beautiful fertilized little nugget. Then you let that grow up for a little bit, and then you implant it in the uterus of the gestational carrier. The gestational carrier is someone who maybe also donated the egg and maybe didn't. Maybe it's someone who just, received that implant from someone else's egg and sperm. And then it grows up in that individual. And then through C-section or vaginal delivery, you have a beautiful baby. But that baby usually shares the same DNA with the egg donor and the sperm donor, not necessarily the gestational carrier. So I was like, maybe that's what's up.
So maybe they mixed up the embryo. Because presumably she would have known that she would have had the IVF, right?
Right. So I was like, oh my gosh.
That would have sucked.
Yeah. So I was, especially because IVF, they're so careful. So I was like, what are the odds? And if you go to the next slide, you'll see that the odds were very low because she didn't do IVF.
Oh, okay.
She conceived spontaneously without any medical support, which makes it even crazier because you're like, then how? Because where else would the eggs have come from? The eggs came from her body.
We had some in chat mentioning possibility of chimerism as a hypothesis.
Save that thought.
Save it, y'all.
Thought. You should definitely save that thought and also be really proud of yourself for having that thought.
There we go. Okay, so possibility of adoption.
Yeah, so this is another theory. This is a theory that a lot of folks in the government policy world were kind of proposing. Was oh, she just adopted them, so she's not biologically related to them. And this is what a pedigree would look like for adoption. And genetic counseling, we draw what we call pedigrees, and they're basically just family trees that dictate relationships. So these straight lines here all suggest a biological or DNA-based relationship, and the dotted lines represent a familial relationship that isn't grounded necessarily in biology. So this would be if Lydia had adopted children of her partners with another person. But again, if we go to the next slide,
we have a question about the shapes. So how does one read this? How does one read this kind of graph?
Yes. These, I love making these. I think they're so fun. So typically you're going to see a square for males and you're going to see a circle for females. Typically this is cis males and cis females. You can also see a circle with AMAB underneath it, and that would be someone who's a trans woman. The AMAB stands for assigned male at birth. And you can see a square with AFAB underneath it, which is a trans man who was assigned female at birth. And also to represent a variety of different patient identities, we can do a variety of different combinations as best suits folks. We can also do a diamond, which is a gender non-disclosed, or for multiple individuals. If you want to say that they're 8 siblings, but you don't want to draw them all out, you'll just do a little diamond and you'll write an 8 inside to dictate dictate that there's 8 folks. The little lines, so you see the line connecting Lydia and the square, that suggests that there is a union between those 2 people, a partnership. Typically, often we will draw that if they have children, or you can draw a line underneath it, like you see between 2 and 3, and then the two children, the circle, the square. And that's saying, these 2 people had these 2 kids together and these 2 kids are full siblings. But then the diagonal dash slashing between 2 and 3 says these 2 had a union that resulted in these 2 children, but they are no longer together. 2 is now with 1 and 1 adopted the children of the union between 2 and 3.
The dotted lines usually included here and it's just that's the adoption takeaway. And then the slash is previous partner.
Correct. Yep. The dotted line means that there's an adoption out of the sort of blood DNA family that you see there, and an adoption out. Because we still want to make sure that the family trees we draw should represent all of the relationships. Those that are DNA based for the sake of genetic testing, those that are biologically based for the sake of any other health concerns we could see, and those that are purely emotional and political and legislative and all of the things, because those also tell us who's interacting with who's having an emotional bond with one another. Yeah.
And then we were folks wanting the square brackets.
Yes. The square brackets dictate just like where the little dotted lines go to. They're just a drawing convention.
Gotcha. And then the final question we've got now is, what kind of data would you use this for? So like maybe just in general, if you have something like this, what would be the functional purpose of having a data set like this?
Yes. No, that's a super, super good question. And I see Sheehan and Deuce. I'm not sure how to read that. I don't know how to read anything. That's a great question, and it's not silly at all.
Ace and Deuce?
Ace and Deuce. Wow, I'm so silly. I was like, is it a play on ocean?
Oh, no. Helen, you should hear me pronounce words.
No, that's so good. That makes so much sense. Okay, well, I love it. But it's a great question. What we use this for is when you go to see a genetic counselor, a lot of times the first thing you'll do what's called contracting, which is basically the genetic counselor will say, like, here's my plan for today. I was going to do these things with you, and then these would be kind of our takeaways. Does that sound okay? Is there anything that you really want to do that I haven't mentioned to make sure that like you both get what you need out of that session? Because genetic counseling is all about like helping the patient. Like it's about what you need and what you want. And then a lot of times the next step will be taking a family history where we draw one of these diagrams. And the reason we do that is because usually folks are seeing us because they have a family history of cancer, or they want to learn more about the genetics of their pregnancy, or they are concerned about their own risk for a certain inherited condition. And by drawing these out, we can see patterns in the family, because typically families share genetics. Sometimes seeing certain patterns going on in the whole family can give us more information about what we think might be affecting one specific individual than just learning about that individual in isolation. And also it can be helpful because it can help us determine who's going to be the most informative person to offer genetic testing to first. And if genetic testing were to come back positive or negative or uncertain, who would be the next most informative people to follow up with and do additional testing.
You're trying to track out potential disease states and or prevent them even. So, right, you can do this ahead of time too, before you have kids, to look at your lineages and see, okay, what do we need to screen each other for to ensure that our kid doesn't have this, or that could be something as well.
100%. Yeah. Like for cancer genetic counseling, for example, if you go into genetic counseling and you say, hey, my mom had breast cancer at 50, your genetic counselor is going to be like, oh, in isolation, breast cancer is the most common cancer that affects, women or folks who are assigned female at birth. 1 in 8 of those folks are going to have breast cancer at some point in their life. That's about a 13% chance. So one family with breast cancer, sad, personally upsetting, but not medically concerning for an inherited predisposition for cancer. If you then add, and my sister had breast cancer and her mom had ovarian cancer and my aunt had breast cancer at the age of 30 and my other sister, like once you see patterns, it becomes more suspicious. And like you were saying, Balint, too, it's helpful for family planning purposes to look at both sides of the family and be like, what are things we should be aware of?
And it's easier to see those patterns with one of these graphs, right? Like if you were to look,
yes,
I don't know if this is still a convention, but it's that you're looking for a certain phenotype. Is it still you color in the circle, or you half color in for like if it's recessive or not, so you can track like, okay, like you can get a lot of information basically from this kind of graph.
Yeah, 100%. So if someone's affected with something, we'll shade in the whole shape or part of the shape, and you'll have like a little key on the side that says like, oh, this pattern or this color means, this condition. This color, this pattern means this condition, and all those things. It allows you to learn more about the chance that a certain family member or multiple family members could be affected with the same condition or different conditions.
Maybe, right? So we have this mom and she has 2 kids with a papa. And
papa, yes.
The dad is definitely their dad.
Yep.
The kids are definitely siblings.
Definitely.
But she's not the mom based on this genetic map that we're leading out, or the sequencing that they did.
Yeah, based on the DNA they did. Yeah. And so now they're coming up with theories for like, what could explain this? They were like, maybe it's an IVF mix up. Nope. Her children were conceived without medical support. They're like, maybe they were adopted. Once again, nope. If we go to the next slide, you'll see that's a no, because she carried them. They are biologically hers. She birthed them. So the other theory that I had was maybe this is a lab error. Like mistakes happen. Maybe this is just like a mix up at the lab. But if we go to the next slide, because they did the test 3 separate times using a cheek swab, which is a reliable form of DNA, cheek swab is going to collect white blood cells as well as epithelial or cheek skin cells. And they got the same result every single time.
So here's a question for you, Helen. What would be a lab error that could or would normally happen? Like what is something, just for the folks to like think about, like what could be resulting as a lab error? What should you pay attention to when you're doing a lab error?
I think my only concern would be like if they mixed up the samples and they like labeled samples incorrectly, or if they didn't get enough of the DNA and so like they didn't have sufficient genetic material to run the test, although that would usually result in a no-call result. So they'll just say like, we couldn't do it because of like testing error. So, I mean, lab error is kind of a stretch. Like labs are pretty good, especially if they're CLIA certified.
I don't think that commonly would come through, right, as something that could be an issue to think about, but it's like, yeah, they check for that, and they did it a few times because I'm sure she was convincing, like, I gave birth to them. We have records. There was a doctor on hand pulling them out.
Yeah. And actually, if you go to the next slide, you'll see, did that help? Did people understand? Yes. So yeah, she gave birth to them, but they don't appear to be related. And yeah, implanted eggs, that's a great. So we explored IVF, but nope, wasn't IVF. And so Lydia's obstetrician, which for folks who haven't been to an obstetrician, is just a doctor who specializes in pregnancy and newborns. They'd been present for her deliveries and her prenatal care and vouched for her parentage. They were like, I watched her be pregnant and have these children. But the DNA still showed that her genetic makeup did not match her children. And because she had filed for support and claimed her children's dependents, she was actually facing like threats for welfare fraud and the removal of her children.
So this was real. So, like, this happened.
This is real. This happened in 2002 in Washington State. Like this is real. And because like, it's one of those things, and I think it's important. I mean, we'll get to it, but I think it's important that we always remind ourselves that genetics is true and science is real, but it's also really, really complicated, you know? So it's I think this is just like really cool.
Where even when we were going through like all the step-by-step bits that we would check, and the science is correct, but we have no idea what it is, right? That's going wrong.
Right. Because like her DNA didn't match her children. And we know that typically children should have DNA that matches the people who contributed their egg and sperm to them, but they're still her children. So we have 2 scientific truths that seem to not match one another. So it's going to be fun to figure out how they match.
Be fun. She tells the father, you cheated on me. But he's like, what do you mean? How?
Yeah, honestly.
That's not usually the direction that this goes.
You cheated on me and then impregnated me with another woman's child.
Because they're not twins, right? The siblings aren't twins, to point out. So this
they're
happened
not twins.
2 separate instances.
2 separate times. And if you go to the next slide, it didn't happen 2 times. It happened 3 times.
Hold on now.
Lydia was actually pregnant with her 3rd child by the same man.
Oh, no.
While this case is going on. The whole time this is going on, she's pregnant.
And so naive question. Presumably they did a blood test on the embryo. Did it not match mama?
So they didn't do a blood test on the embryo, but after, so she delivered during the case and the judge ordered a court official to be present at the birth. So like she's delivering and there's like a person there in a suit holding a briefcase. This is my headcanon. I don't think he was holding a briefcase. In my headcanon, he's in like a 3-piece suit, double-breasted. That's what's happening.
No.
I hope so, though. And as described in the Embryo Project, which is one of the sources for me putting together this presentation, the officer watched Lydia give birth to her 3rd child. The doctor took blood samples from both Lydia and the baby, ran them in the lab, and Lydia once again came back as not genetically related to the child that she just gave birth to.
Oh. that's a toughie right there. Yeah, because even the invasion of privacy, on this, right? Of like I get like you get it, but like at the same time, it's like, oh, the poor woman who's just trying to give birth to her 3rd kid is being watched by an official.
Yes. And like the stress and the strain and like having to prove something that you never thought you were going to have to prove, like, is just wild. Yeah. And okay, I see a question from, history of leukemia and a bone marrow transplant from her husband. That's a great question. My mom actually asked that because I was talking to my mom on the phone. I was like, listen to this cool, crazy case. And she was like, obviously she had a bone marrow transplant. I was like, okay, not obvious. And also no, but also really, really, really good guess.
So if she would have had a bone marrow transplant prior to having any kids, right? So that would be a critical step because then the blood that the kids would have been Lydia's original blood type, but her bone marrow would have been a new blood type, which was a given from the donor, right?
And that would also assume that they'd tested Lydia's children and her with blood, but they did multiple sample types. They did hair, they did skin, they did blood, and it was all coming back the same, but not related to her kids.
It's really cool. It's really, really cool. It's a good puzzle.
Yeah. No, it's wild. Okay, go to the next slide. It's very fun. I mean, scary and terrifying. Like, it's fun now for us in hindsight, but for Lydia, this must have been like a nightmare. So I do want to be like conscientious of the fact that like now we're like, oh, what a cool scientific phenomena. But like in the moment, just like god awful on like an emotional scale.
Yeah. And also just the next slide, the lawyer had an idea. Like, why does it have to be the lawyer who has to have the idea on these things?
Exactly. Like, well, and also Lydia had a really hard time getting a lawyer to even take her case because everyone was like, you're going to lose because DNA is law. Hello, cutie.
Oh my gosh, is that a hedgehog? She has to wipe my mouth from dinner. Yeah, she had to wipe hers and had to do mine too. Yeah.
Very thoughtful.
Thank you, Munchkin.
Very thoughtful.
Yeah, so it's like the lawyer comes up with this trick, and that's just, it seems like something that the lawyer should be needing to come up with, right? That's, to me, almost seems like we just had someone watch.
100%.
But even actually,
and like, her doctor is vouching.
Honestly, even if the judge ended at that and he's like, you know what, the heck with it, let's get funding, I would still want to figure out, well, like, what's going on? Like, and is this something the kids should be concerned about? Yeah,
because the court was concerned that it was like surrogacy fraud or like something like that. But I agree with you that like, I think me as a parent, you know, I would be like, is this okay? Like, are they, like, where are they coming from?
Yeah.
Like, if they're not, if they don't share my DNA, like where, and half their DNA comes from their dad, where's the other half coming from? Because it's got to come from somewhere.
Yeah.
So Lydia's lawyer had an idea. Okay. So following the birth of this child, he had an idea, and scientists tested this hunch by running a DNA test using a swab of cells from Lydia's cervix. So they hadn't tested this cell line before. Your cervix is part of the gynecologic system for folks who are assigned female at birth. And previous tests had been on different kinds of samples. So this was like a brand new kind of tissue.
So
what do you think the result? Oh, yes.
Why is that unique cell type unique from the cheek swab or even, I guess, the skin biopsy? Like what makes that unique?
So for most people, it's not. For most people, it would be the same DNA.
Okay.
But the reason that they wanted to test the sample type is that it's closer to your ovaries, which is where the eggs or the germ lines develop for folks who are assigned female at birth and have ovaries and a uterus and a cervix and those parts. And so the thought was just that they're closer together. And so it's a greater chance that those cells share a common origin.
I see. That's really interesting.
Yeah.
Okay. Makes sense.
I mean, wild. And once again, that this poor woman is going through all of this with a newborn and 2 young children at home.
Yeah. and a very concerned husband now.
And then her lawyer's just like, I'm gonna need one more sample, and this one's a little invasive.
I guess one question that folks are having too is what about the mitochondrial DNA? Has that been tested between kid and mom?
Mitochondrial DNA? Hold up. That's a really, really, really great question. I hadn't thought of that. So for folks who aren't familiar with mitochondria, they are famously the powerhouse of the cell. And mitochondria live inside each and every one of our cells for the most part. I'm always doing this. As you know, science and all of its caveats, we always go in each and every one, usually. And it's like, but in the vast majority of your cells, you have mitochondria. And that's because mitochondria are like the generators for your cell. They're how your cell gets energy. But what's cool about mitochondria is that it's maternally inherited. So you get your mitochondria from your mom. So you and your biological siblings that share a same mom, the same person donated their eggs to you, they are all going to have the same mitochondria, but the sperm does not contribute any mitochondria to the embryo. So it's matrilineal inheritance. One of the theories I heard one time, I don't have a source for this, but one of the theories that I heard one time in grad school was that the mitochondria and the sperm is like all locked into the tail, and the tail doesn't actually fertilize the embryo. The sperm kind of just like latches onto the egg and like yeets its DNA into the egg, and then the rest of it like dies. And so, I mean,
you're right. I love that.
It's just like the DNA gets in there, but like that's all. It's like just the raw chromosomes, and so all the mitochondria and like the proteins and all like the nutrients is in the egg. Yeah, which is crazy and super cool.
Yeah.
But no, they did not test mitochondria, or I did not read about them testing the mitochondria. But the mitochondria theoretically would have shown that They were, but the thing about mitochondria that's tricky, so I'm thinking out loud now. The thing that's tricky about mitochondria often display what's called heteroplasmy, which means that mitochondria have their own DNA separate from the rest of the DNA in the cell. And that's because we think that mitochondria formed when like one organism, like a bazillion million years ago, that's not a real number. I don't know how long ago it was, but many, many have passed. And one of those organisms consumed another one. And normally it would like digest it and eat it and it would be great. But this time the little one was like, what if I hung out in here? And the big one was like, I guess that's fine. Can you make me energy? And then it became mitochondria over time. Because that mitochondria have their own DNA separate from the rest of the cell. And usually multiple mitochondria in the same cell will have different DNA from one another. And so even though you inherit all of your mitochondria from your mom, you might have different mitochondrial DNA because of that heteroplasmy, because maybe your mom has, 10 different kinds of mitochondrial DNA and you happen to inherit like these two and your siblings happen to inherit like these two, So I think it's a little more complicated because of that heteroplasmy.
So
does that make sense at all?
If you get a no on that, it's not a conclusive no. Is that fair to say? But if you were to get a yes, then that would be a conclusive yes. So it's a squishy answer.
I think that would be my guess. I think my guess would be that it would be like squishy, unless you found that mom had like a high degree of homoplasmy, which means like a lot of mom's mitochondrial DNA is very, very consistent. It's like the same. Some genetic conditions are inherited that way with homoplasmy, but heteroplasmy is the norm. Mm-hmm. So it would really
because it's all mixed up, the likelihood of being a perfect match is even lower. Yeah, that makes sense.
This would be my guess because there's more diversity in that DNA.
Yeah, we had a question from Chad too. I think this was referring to the yeeting fact. Does the yeeting fact contribute to how they do the 8-week male-female test via blood?
Oh, that's a very, very cool test. So that's called NIPT, non-invasive prenatal testing, or non-invasive prenatal screening, or cell-free DNA. And it is different, but that's very, very cool. It's popular because it's a way for couples to learn more about the genetics of the pregnancy, or for pregnant people to learn more about the genetics of the pregnancy prior to the 20-week anatomy scan, which is when they usually take the pictures and they count all the fingers and toes and they look at all the stuff because the fetus is usually big enough at that point to get some really good pictures. But folks like to learn about information a little earlier, and that's with this other test called cell-free. The way that test works is that we know that during pregnancy, naturally, there's the fetus, there's the placenta, and there's the gestational carrier. And we know that naturally, some of the DNA from the placenta, which connects the fetus to mom, washes off into mom's bloodstream. Totally normal, totally natural. But that DNA is now in mom's bloodstream, and it's not kept safe inside one of the cells of the placenta or one of the cells of the fetus. And so it kind of fragments and breaks apart because it's not being protected. So we call it cell-free DNA. And so what we know is that by about 10 weeks gestational age, about 10%, it's going to vary between pregnancies, but about 10% of the cell-free DNA in mom's blood is actually going to be derived from the placenta. And the vast majority of the time, the placenta and the fetus have the same DNA. So by taking a sample of mom's blood, we can look at the DNA present. We can parse out what's mom's and what belongs to the fetus or the placenta. And then we can learn more about the genetics of the pregnancy by assessing the genetics in that cell-free DNA derived from the placenta. It's the same kind of like technology that they're assessing the DNA with each time, but it's different sample type. So it comes from a different part of Lydia's body than they've tested. And it comes from a cell line that is closer in proximity to the germline cells, the egg cells. Because basically they're like, we know that her children came from her eggs, but her egg DNA doesn't seem to match the other DNA we've tested. So can we find any DNA in her body that matches the DNA in her eggs?
And so this proximity isn't due to adult proximity, maybe. It's due to during development of the embryo. The cells that give rise to the ovary are the similar or lineage cells that give rise to the egg as well, right? Versus like the brain is a little bit further differentiated. It might be different in proximity in the embryo. So this is taking a look at developmental biology of a human.
Yeah, that was the theory. Also, this you baked those buns personally bit is very funny to me. This little comment right here from Mara's teeth, that's very funny to me. But yeah, no, so that's exactly the theory that they had.
Oh, Morris also introduced me to the concept of saying yeet to throwing out like organs as well. Morris,
great.
Helen had said like when there's fertilization of the egg from sperm, it's because the sperm yeets the DNA into the egg. Morris, I couldn't help but give a thing to you on that when you taught me about eating organs as well.
Because I mean, it's obviously more complicated than that, and it's actually like the process of fertilization is actually really, really cool and complicated and fun. But yeah, they just like the DNA does eventually just end up in there, but it's crazy. It's
and not the mitochondrial,
and not the mitochondrial. Yeah, mitochondrial is all from the egg.
Yeah, it's a brilliant system, really.
What do you think happened this time? Do you think it was the same? Do you think it was different?
I think it was. I want to say it was a genetic match.
Okay, to all 3 children?
Yes, let's go with that. Chat, what do y'all think?
Hell yeah.
I'm going to go all in. What do y'all think? Was there if you sampled these cells, the cervix cells, which are closer in development to the rest of the developing embryos, like as Lydia herself was baking, right? It would predict that it would be closer. What do y'all think on that front? Thinking that I have no clue, says Ace. Wombat says 2 out of 3 match. Alex is like, I already know this story. I ain't going to vote.
All 3 because no more shenanigans.
All right, let's we're going to go in all 3 and we're going to lose all of our money, I imagine.
Next slide.
Oh,
this is the paper that made the lawyer suspect that Lydia could be a chimera and therefore request the chief testing. So someone said this right off the bat, which we love. So a chimera in Greek mythology, I'm pretty sure is like part lion, part eagle, part snake or something. Part goat. Is there goat in there? Is it goat, lion, snake?
I think it has a goat in there.
Because griffin is lion, eagle. Hippogriff is eagle, horse. Anyways, okay, part lion, part goat, part snake. Thank you.
There we go. Yeah.
That feels right. But a chimera in a human person is when there's one human being who has 2 genetically distinct cell lines. So 2 sets of DNA in their one body. Most chimeras don't know that they're a chimera. They don't know that they have more than one type of DNA. And actually scientists these days are thinking that it's more common than we used to think. We used to think it was this like crazy rare phenomena. And now some folks suspect that it might be as common as any case of fraternal twins, which would be about 1% to 2% of all pregnancies. So if we go, oh yes.
So is this the paper that was on Lydia, or this is a different paper that was like informing the lawyers of it?
Correct. Okay. The second one. This is the paper that the lawyer was like, in my headcanon, again, I don't know if this is what really happened, but in my headcanon, the lawyer's like tippy tapping on his Google and he's just like, how can a person not have the same DNA as their child? And this paper popped up and he read it and he was like, that's crazy.
So what we know so far is just that methodology is that there's 2 different types of DNA sets in a person. Are there any rules that govern that kind of output? Like what are the inputs that what how can that manifest? Like what are the different variances? Or will we get into that in a little bit?
Yeah, we'll get into that. Okay, so if we go to the next slide, we'll kind of talk about how chimeras happen with people. So we go back to the beginning, and a singleton pregnancy, vast majority of human pregnancies, is when you have 1 sperm, 1 egg, the 2 shall meet, and then they develop into an embryo, which implants, grows into a fetus, grows into a baby's born. Every single cell in that baby's body has the same DNA, half of which comes from the egg, half of which comes from the sperm. Amazing. Sometimes, if we go to the next slide, you can have identical twins. Identical twins is when you have the same process, 1 egg, 1 sperm, they fertilize, but very early on in development, that fertilized embryo splits into 2, and it becomes 2 embryos, which then implant separately and grow into 2 distinct fetuses that are born as 2 distinct people. But these 2 people have the same exact DNA in all of their cells and as one another.
Because they had 1 embryo fertilized that then split. So all the genetics were all in that original embryo.
Exactly. And what's important to note here is that the embryo splits, not the cell. So they both still have the right amount of DNA because the embryo is composed of many different cells. And so when it splits, some of the cells go here, some of the cells go here to the different fetuses, and then they divide and continue to grow properly.
And it's early enough in development where you still really haven't had like specifics in the cell types. It's just you're a cluster of cells.
Exactly. Yeah. Yep. You can also have fraternal twins. Genetically speaking, fraternal twins are basically the same as siblings, but it's just siblings that are born at the same time. Siblings that are like womb mates. They share a uterus while they develop. So it's caused by hyperovulation. Hyperovulation is when instead of the ovary yeeting out, as we would say, 1 egg at a time, it yeets out 2 eggs. Both eggs get fertilized by separate sperm. So you have 2 developing embryos. Both of those embryos develop into a fetus. Both those fetuses develop into a baby when they get born. Each baby has all the same DNA in their bodies, but their DNA doesn't match one another. So that's a fraternal twin. And then there's chimerism. Chimerism is when, let's imagine we've got fraternal twins going on. So we've got 2 eggs, 2 sperm, 2 developing embryos. Sometimes chimerism can occur in cases of vanishing twin, which is where one of those sort of embryos doesn't implant, doesn't start to grow into a full fetus. but some of those cells get absorbed by the embryo that is still growing. So you end up with a fusion, not unlike how identical twins are split. Instead of being a split very early on, this is a fusion very early on. And so then you get the development of a person who has two distinct genetic cell lines, even though they're just one person.
So that doesn't ruin the embryo because it's so early on that you're still just a cluster of cells, right? You're still going to be getting information of this. You need to become this cell type. You need to become this cell type. And that's not dependent on the genetics like being different because there's not like, they're not cycling and recognizing that your genes are different. You're just getting information of what to become.
And the embryos are fusing. So like the cell might join, some cells might join another embryo, but in the same way that with identical twins, each individual cell still has the right amount of DNA. In this case, the embryo might fuse, but each individual cell still has the right amount of DNA. So exactly like you're saying, unless there's a reason that the cells wouldn't succeed, either based on genetics or some other factor that would cause the pregnancy not to take, then if both cells have genetic information that is compatible with life and compatible with one another, then there's no reason that wouldn't be a perfectly healthy outcome.
And because presumably during the embryo, like you're getting like cellular gradients telling what to be turned on and off. And so it's all that those other cells can still read that information and get that information in, right? So that you're not at any kind of weird loss. You're still fine in terms of functioning, but there is a limit, right? Like of when that fusion can happen, like late enough in development, presumably it's not going to happen. Like, or actually, I guess as Wombat says, if you go late enough in development, you might have a conjoined of two twins growing together, but that's very different than something like this, right?
Right. Absolutely. Yes. Yeah. Definitely like the developmental stage and the reason for why you would have sort of one embryo absorb into the other. Like a lot of factors are going to influence what the outcome looks like. But in the case of Lydia, if we go to the next slide, we'll see that what she had actually was a very small subset of her cells, really just those in her eggs and parts of her cervix, maybe other parts they didn't test because you can't test all the internal organs for their DNA, for example, were one cell line. And then the vast majority of her body was the original embryo. So this might have been a case of like vanishing twin, where very, very early on in development, one of the embryos didn't make it. And so a few cells got absorbed by the other embryo that was developing. And so they became parts of Lydia's body, and the other cells became the majority of Lydia's body.
So this would mean that not the entire secondary embryo was absorbed. Is that fair?
Or just that, like, yeah, that would be my guess. Yeah, that like some of those cells like just passed and weren't absorbed. Yeah, but then some of them were, or maybe for some reason like one cell line dominated over the other in these early stages, and then just like it was a numbers game. Yeah, that would be my theory, is that It was just like a numbers game, or because I can't imagine why there'd be like an evolutionary, well, unless there was like some reason that like the cell line that was in the majority of her bodies was more rapidly dividing at that early stage, but she didn't have any health problems, or her children didn't have any health problems. There's no reason to suspect that the more rare cell line was in any way less genetically fit than the other.
Gotcha.
So yeah. Does that make sense? I was kind of thinking out loud there.
Yeah. Someone had a question, Helen, earlier in the chat. This was interesting, is why wasn't there at least some match, given this could have been an aunt?
Oh, sure. I see what you're talking about. Like, why wasn't, because, yeah. Okay, no, I see you. That's good. It's a really good question. That's a really cool question. Yeah, because the DNA would still be from the same grandparents. Mm-hmm. And so you would expect overlap. It might just be that there wasn't the kind of genetic overlap that they would expect for maternal inheritance. And so they may not have reported back the kinds of details or gotten the kind of information that would allow them to confidently ascertain the kind of relationship that would be from an aunt. They might have just said, like, it's definitely not maternal. And so that might have been, without looking at the exact results and knowing what kind of testing was done, I wouldn't know if the kind of testing that was done would be able to confirm that kind of relationship, if they were purely doing like maternity-based, like parentage-based testing. Because there are different kinds of genetic testing. So I'm not sure what kind they would have done and if it would have had that sensitivity.
Or maybe they just happened to pick a locus that was really variable. Right. And so maybe that's intentionally, like, you just pick that zone of the genome that's going to be vary enough where even the aunt level variance would be enough to be like, this isn't your kid, and that's the extent of what you're looking for, like you said.
Yeah. That's a really good question, though.
It's really cool to have just like, it makes more sense, I guess, easily, like the 50-50 chimera. But then this kind of like ranged chimera that gives you positional information on where it was absorbed is really, really cool.
Right. Yeah. And also, like, I think people used to think too, right, that chimeras, you would always know, like you would have like very different, like you would know. But I think what we're learning more is that a lot of these folks don't know. Like a lot of people don't realize that they're a chimera, in the same way that the more we do genetic testing, the more folks are being incidentally diagnosed with certain genetic conditions that you wouldn't have tested for because they didn't have health problems, but they're being tested to learn more about their pregnancy, or they're being tested for a family member. And then they find out something about themselves that they hadn't anticipated.
So would you say there is an increase in the rate of chimerisms that we're aware of because there's been more testing? And what if anything, like if I get tested and it's like, oh, you're a smidge of a chimera, does that affect me in any real way?
I think it would depend on like why you're being tested, right? Because like if you're being tested for a health concern, maybe this could explain your health concern, because maybe some of the chimeric, one of the cell lines carries a genetic predisposition for something. But if it's incidental and you don't have any health findings, I don't think if both genetic cell lines, this is me thinking, this is not me knowing, I would think that if both cell lines are healthy and you don't have any health concerns and you're a full grown person and you have healthy children, then all is probably well, would be my guess.
That's really interesting. So it could matter important. I wonder too, like if you were to, if you are getting tested for like potential, like having kids, the importance of knowing that you could be a chimera and where you would be tested, like where you'd be getting the samples from, seems like it could be really, really important, right? Like you were saying,
yeah, absolutely. And we do always also, like if someone comes back and their genetic test results imply that there's like homogeneity or heterogeneity or differences, a combination of DNA in their tissue, you would already be suspicious. But we do always quote, even if a person, has a blood test, a saliva test, a cheek swab, the most common DNA samples, and it comes back, this is your DNA, you only have DNA from one cell line. We don't always include the caveat of like, you could be a chimera. But for reproductive purposes, we do always include the caveat of something called gonadal mosaicism, which is just the argument that your egg cells or sperm cells, but typically we say egg cells because typically folks that are born with ovaries and eggs, you're born with a finite number of those, and they all developed while you were developing in the uterus in which you developed. Whereas typically folks that produce sperm, the sperm has a higher turnover rate. And so we don't worry about mosaicism as much there. But gonadal mosaicism, we always quote too, because there's a chance that your eggs might have different DNA than you do. So maybe somewhere in the development of your eggs, a genetic change occurred only in those eggs. So you don't have any of the features of that genetic condition, but there's a small chance. We usually quote about a 1% to 2% chance of gonadal mosaicism. It's probably lower than that, but we usually quote that number. At least I learned that in grad school. Things might have changed in the last 5 months or however long ago it's been.
So No Talent Idiot Writes Music, which is the handle, that is not me imposing anything. I'd asked the question of should that manifest as a mosaic pattern in the body cells? And I guess it did, right? But it was in only certain cell types that they saw this mosaicism. It wasn't like the image that we were looking at here with the baby, where it was like a 50-50, like there were just, dots throughout of like different colors. But for here, it's like you'd only have it in particular tissues, right?
And so I think that's the way it could be. It would just depend on the way those cells developed. So like there's a theoretical possibility that one arm maybe could have DNA from one of the cell lines and the rest of the body could be different. Or it could be half and half, or it could be woven in between, or I think it would really just depend on the way those cells grew. Again, I'm not a, specialist in chimerism or embryologic development, but I mean, I think that there's all those possibilities. And actually, fun fact, so folks that have 2 X chromosomes, so most women, folks that have 2 X chromosomes, during development, something happens called X inactivation. Balint, you've heard of X inactivation?
The beauty of having the 2 X's, but then some zones are turned on and off. Yeah,
exactly. Because after development, your body's like, wait, I actually only need 1 X's worth. Like after a certain phase of development, your body's like, I don't need both these X's. So they randomly start turning 1 off in every cell, but they turn them off differently in each cell. It's not like they're all like, let's all turn off the copy that was inherited from the sperm, or let's all turn off the copy that was inherited from the egg. So it's random. So like in my body, for example, because I have 2 X chromosomes, half of my cells roughly have the X inherited from my mom and half roughly of the X inherited from my dad. but it's not like my left half is my dad's X, my right half is my mom's. It's like potentially random, or it could be more, lineage derived. Like maybe my skin cells are from my mom's X and my, guts are from my dad or whatever. Who simply knows? I have not tested, but yeah. Or calico cats. Yeah. Calico cats are, I think, always female because it's the result of X inactivation resulting in that color. I think. I don't remember. I studied human cells.
Yeah.
But I think.
I believe it. Right. I think so. Yeah. And then can an individual get both the male and female gametes? Asked Wombat.
I meant to look into this and then I didn't because the rest of my job was calling me. I meant to look into this before today's chat and it never happened. But I was also like, what would happen if one of them had XY and one of them had XX chromosomes? What would that do? What would that matter? Because I don't know if it would matter depending on the stage of development. If the gonadal sex has already been determined, hormonal sex has already been determined, I don't know if it matters. Because I know that with blood transfusions or bone marrow transfusions, you can get a bone marrow transfusion from anyone of any sex. If I needed a bone marrow transfusion, they wouldn't only look for other people with XX chromosomes. They could give me a transfusion from XY. So intersex, there's a great question in the chat. I'm not an expert on differences of sexual development or intersex identities, but I know that intersex is very complicated, and I do not think everyone who is intersex is a chimera.
Or
I think that would be an oversimplification.
If you were to get a blood transfusion from someone who is XY, if they did a karyotype then on you from your bone marrow or a derived cell type from the future, then you would come up with the karyotype would be different from other cell types that were not bone marrow derived.
Yeah. My skin and all of that would still be XX, my hair, but my blood cells would be XY.
Also be really cool. Just as an aside of you have two different karyotypes in you.
Yeah. No, it would be crazy. Yeah. And that test that someone mentioned earlier, that cell-free test where you can learn more about the pregnancy's genetics, sometimes you incidentally will learn something about the person carrying the pregnancy. So sometimes those folks will incidentally find out that they have, triple X, for example, which is a genetic condition where you have 3 X chromosomes instead of 2, or, some other genetic condition. So we do always warn folks when they get this test done that it can give you helpful genetic information about their pregnancy, but also you could incidentally learn things about yourself.
And that may or may not affect the pregnancy.
Right.
Yep.
Right.
So what does this mean for Lydia, this chimera?
So for Lydia, basically, this is the end of our story with Lydia. If we go to the next slide, we can kind of transition to another case of chimerism if folks want. But basically, all this means for Lydia is that she's a chimera. They're basically fun fact for you to know, you are allowed to have your children. Sorry about all of the trauma. Is basically kind of what happened. So they wrote about it. Her case is included in quite a few different textbooks about how the law deals with DNA. I think there was a documentary that was made that she was in that I did not see, that I think sort of featured her story, because it is always all very interesting. Yeah.
That is such an underwhelming. Sorry. A lot of trauma there.
Very underwhelming.
Hope you're good.
It's hard to be you can also, it's crazy that her reward is the absence of a bad thing. They're not here's a good thing for all the things. They're we won't take your kids. And it's they never should have based on DNA.
Yeah.
So it is wild. But yeah.
I kind of hope that she countersued and made bank.
I have no idea what happened. Yeah. Because because there's so many, when in doubt, we should always assume best intentions, right? Always assume everyone is trying to do the right thing. And so in my mind, they weren't trying to be obstinate, the courts. They were trying to make sure that there wasn't surrogate fraud, that she wasn't, kidnapping someone's children.
Sure.
I understand the intention, but also at a certain point, you're I think for me, but maybe also because I believe that science is so complicated, and part of the scientific mindset is always being open to new data. I think to me, once the obstetrician, but maybe I'm also too trusting and I just believe people. It's once the obstetrician's vouched for her and she has birth certificates and everything, I feel at that point, I'm something's up with the DNA.
Yeah. It's being able to be accepting of new things.
Right.
Without, maybe, I don't know, I'd be naive too, but without questioning you sent a lawyer in to watch the birthing, right? So you'd assume that.
Right.
That's when that test comes back negative, how do you explain that away? Unless you're suggesting that she's stealing embryos, from the closets in the IVF facility, but then self-injecting. I don't know.
Yeah.
I really, really thought the story behind the non-mom, but actually being the mom was wild.
I know.
And it was a really, really cool take, because it not only talked to us about like basic genetic testing, but also like how chimerism worked. It had developmental biology put in there. It had all the different features pulled in.
Legal implications. It's a crazy one.
It was really, really cool.
Thank you guys so much.
And Helen, thank you so much for your time.
Of course.
We, as predictably, went over, and so thank you.
We cannot shut up.
No, thank you for staying with us and chatting the beautiful science that goes there as well. Thank you so much.
Thank you so much for having me, for having Fred Hutch. Thank you.